told earlier, but unfortunately it was not possible. So what I'm going to do is talk about a few of the model organisms. We have a few model organisms when it comes to in genetics and in as well as what do you call it in life sciences, whose role is very important as to for us to know different kinds of for basic laboratory studies. So what is it that we're looking at? We are not going to earlier there was a time, especially during World War II, wherein human subjects were used with or without the concern. That is a different case altogether. And wherein they were used, they were tested, they were the lab rats, as you can unfortunately call it. So what is a model organism that we are looking at? It is the most suitable organism to study a particular disease or a phenomenon, or to see as to how it is going to react, especially for drugs, or how a disease is progressing. And in like because it is similar to that of the human beings. In it is not in vitro, that is the laboratory tested. We are not looking at the what do you call bacteria, but they are in vivo models within another organism altogether, widely used to see human diseases usually, and wherein human experimentation is unethical. So, what are we seeing? We are looking at organisms such as rats, rodent or pig family. Usually, the rat family is very commonly used, and it is accessible and it is quite easy to study. The model, the mouse model, especially. And at the same time, we have certain organisms such as Rhabditis elegans. We have Drosophila, that is the fruit fly, which is the very good model for developmental biology as well as for genetic studies. So, what is it that we are looking at when we are selecting a organism for it to show any kind of trait? We are looking at the size; it should be small, and its development should be short, and at the same time it should be easy to observe and to see its experiment. As such, availability of the genome sequence, and how easy is it for it to be transformed, what are the how easily is the trait being transformed within it, and at the same time is its genome already sequenced, and an array of tools and techniques for whatever molecular genetic studies we are doing. How is is its genome easily manipulated, and are we able to easily select, identify its genes? Also, if you see, for example, Drosophila melanogaster, that is the fruit fly, number of the genome size is small, because of which it is easy for us to actually sequence it, and we know at what locus of the genome we have what kind of trait, because of which it is easy for us to manipulate. So these are the criteria that we are looking at: the small size, development time, amenable to observation, and at the same time, good online base. And in case we see all of this, we even choose bacteria, or we even choose other organisms. So one of the most commonly chosen ones are, in the case of bacteria, are E. coli. It's gram negative, and it is a rod-shaped bacteria, in fact. And usually, where is it present? In our lower GI tract. Of almost all the what do you call it homeotherms, and it is expelled through fecal matter. So E. coli is a very common contaminant that is present in the sewage water, and it can grow massively, especially in aerobic conditions whenever there is oxygen present in within 3 days. And it is really small. The genome of it, the size of it is small, which is around only 4.5 to 5.5 mbps. So it is quite small. We can easily cultivate and at the same time grow. So why is it a model organism? Because it's unicellular. So we do not have any kind of issues with it, and we know its genome. And at the same time, the doubling population is around only 20 minutes, since it is a bacterium. So it can reproduce very quickly, and we do not have any kind of ethical concerns because it is wrong. There is something called as ethics that we have to keep following, especially during any kind of organism being used for any kind of experimentation. For a tumor or a cancer growth which is painful, you need to use there are certain medical codes and ethics that have to be followed. So in this way, in bacteria at least, you can be sure that for growing, manipulating, and for bacteriolysis, we do not have any kind of ethical concern, unlike chimpanzees, or that is monkeys, or for mice also. And research time is relatively going to be lessened, and adaptability for it to grow in different conditions is going to be smaller, you have a larger, I mean, you can grow it in many kinds of atmosphere. And when it comes to the culture media, again, you need not have a lot of culture, expensive culture media. In fact, the basic nutrients are available. In fact, it can grow even in sewage water. And MS media, then the liquid or solid media, whatever it is, agar agar, given with basic minimal essential media is given, that is enough. Eagles media, or for that matter, the MEM, that is minimum essential media, that is sufficient for the bacteria to grow. Liquid culture, again, only up to a billion cells per ml. So that's quite a lot. And trillion can be grown in one single test tube. So we are looking at a huge amount of bacteria being grown in just one test tube, which is again going to be useful for us. And if you see, the cells are diluted, spread into a solid medium, petri dish, and then each individual bacteria, since we're talking about bacteria over here, can give rise to a concentrated clump of nearly up to 10 to 100 million. That is 1 lakh to 10 lakh genetically identical colonies. Once this is done, isolation can be done, and you can easily once the strain is developed, you can screen it, and then you can see, since it's a circular DNA molecule, you have around what do you call it, 4 to 8 protein coding genes, ribosomal operons, that includes again operons, and 86 tRNA genes. So what is it used in right now? You see, it is used as a vector for genetic element synthesis of proteins of interest. And we have something called as, once bacteria E. coli is done, we have a plant over here. That is a plant that is belonging to your angiosperms. It is Arabidopsis thaliana, which is the Drosophila melanogaster. It is one of the most studied, well-studied plant in the whole of plant biology. This is Arabidopsis thaliana. It is a member of the Brassica family, that is your cabbage cauliflower family. In fact, it's a weed. It's a small flowering weed which is growing abundantly for us. And it is quite a lot, more number in size, in fact. And it again has only, it's a very small genome having only 5 chromosomes. So because of which any kind of genetic manipulation can be done in this. So what is it used for? Of course, the immune system can recognize virus, different kinds of any kind of microbes for that matter, insects, worms, and you can also study abiotic and biotic stress that is there in the plants. And it is most widely studied for its genome structure, regulation, development, evolution in the plant kingdom. Also, it gives you a very important knowledge about your plant genetics, and it is applied so that all of this can be applied to the other important plants. And how is it that we are going to see? You can transform Agrobacterium tumefacius, which is into this Arabidopsis thaliana. So what is going to happen? You can actually identify mutants, which is around 1500 available mutants that are there, and through reverse genetic that can be done. You can insert around 1 lakh insertions at different locations in the 5 chromosomes that are present. So it is a wonderful plant. And why is it a model, role model, in fact? because 90% is basically in the whole of biological models, it is plants, of which you have around 250,000 species of flowering plants, which can actually produce around 10 to 40,000 seeds. So, keeping this in mind, Arabidopsis thaliana can also be grown in the lab. So many variants of it again are available, and self-fertilization is possible, and outcrossing can also be done. And around 125 million base pairs of DNA and 18% of which are genes that are common with the human genome have been identified in Arabidopsis thaliana. And its average gene size is around 200 base pairs. So it is the first plant whose complete genome was sequenced in 2000. Keeping this in mind, we have a photosynthesis, carbon and nitrogen fixation, and anything that is especially studies regarding secondary metabolites for nutrition or as medicines, and different kinds of evolutionary mechanisms, and how cell to cell communication in plants has been done, and all of it were identified through plants only, and all of it can be seen in Arabidopsis thaliana. And why is it a useful model plant again? Because of its simple genome, we have only 5 chromosomes, easy to maintain, space efficient, can be grown in the lab. It is its principles are applicable to the other plants as well. Increase of food production, in terms of quality and quantity in the whole of world can be applied by studying just this one plant. And overall application of all such kind of practices through study of only one single plant has made it an excellent model for as a plant for the studying of different kinds of plants. In fact, now we have come to a single celled microorganism that is in the fungus kingdom, that is the yeast. So far we have seen a bacterial rodent model, which is E. coli. In plant kingdom, we have seen Arabidopsis thaliana. In the animal kingdom, we are looking at yeast, which is a single-celled microorganism, and it belongs to the fungi. So, how is yeast used as a role model? Let us just see. For that, we have to know the characteristics of yeast. In this, we will get to know that first thing is yeasts belong to fungus through what do you call it? They have two types of reproduction, that is asexual by mitosis and by a asymmetric division, which is the budding. Also, where do they grow? We have a lot of useful for use of yeast even in our food, what do you call it, bakery industry. In fact, yeast is used as a starter culture when you're baking bread, you're making cakes for that matter, or you're in the fermentation and production of alcohol. And at the same time, you have so many uses for yeast. And yes, it can grow as a single-celled organism also. Yeast can be they grow as just a single cell or with buds. As compared to that of the other fungi, and at the same time the other fungi are growing as molds, and they have hyphae, mycelium, and they're connected to each other, whereas it's not the same case scenario with yeast. And they vary in size depending upon the species and the environment. It's around 3 to 4 micrometers in diameter. Some yeasts you can see it's around 40 micrometers in size, so you can actually see in the naked eye. And it is one of the most simple eukaryotic organisms that is there, and you can compare it with that of the human beings also. See, we have to remember that nearly over 95% of all the genetic sequences in almost all the living organisms are same. It is just that 1 or 2% which is varying, which makes us different from all the other creatures, in fact. So yes, we do share um, our genome. And when you have sequenced, you see that there is a lot of analogy, or between all the organisms, as well as with as compared with that of the human beings. So yeast was the first eukaryotic organisms whose genome was sequenced. So at the same time, when you can compare the genome of yeast and humans, and you have found out or identified that there is a lot of similarity, you can study diseases pertaining to the human beings in the yeast and use it as a model. So we have certain species such as Schizosaccharomyces bombae, which is a fission yeast. It is a popular role model for studying the cell growth and cell division. That is because it's quite, I wouldn't say cheap, but yes, it is not as expensive as other organisms to grow in the lab. And also, they grow only up to a certain amount of length, so you can control its growth as well as cell division. And it has a lot of genomic data also, in fact, that you can see. And there are many genetic studies mechanisms for studying of the genes. So what is it that we are looking at? Through microarray or RNA sequences, you can see the gene expression. And when we are talking about the gene expression, we are looking at the different kinds of the cell cycle: G1, G2, S phase, M phase, all of it put together. Indels. So, how is it working in the case of your, what do you call it, whenever you insert or delete any kind of codons or any kind of, what do you call it, the your codons basically. And then if there's any kind of chemical disturbances, and also how are you able to see what are the transcription factors that are actually causing a regulation. So yes, transcription factors can be studied through your chip sequences, and at the same time through what are the proteins that are regulating the transcription. And basic protein interactions, that is your, we have a yeast 2 hybrid system, in fact, which is known as Y2H. And of course, just to know the complete genome sequence, since we know, we can actually tweak and find out a lot of stuff. And at the same time, you can do a lot of on what do you call it, a basic um, um, any kind of test for that matter. And when it comes to genetic interactions and pathways, you can actually see the synthetic genetic array. And when it comes to the chemical genomics, you can just see basic small molecule gene interactions, insert or delete a sequence and find out whether it is affecting its the product or the protein sequence or the that outcome. And then according to that, you can screen. And it is quite good as a role model for understanding human diseases and human biology. So almost 4 Nobel Prizes just involving yeast were awarded for the different kind of researchers, because of the kind of research that was done, and wherein yeast was involved. And the genome of Saccharomyces cerevisiae was published in 1996, and Bombay in 2002. And you have a yeast genome project, in fact. And at the same time, for all the functions of the genes in the genome had to be elucidated, projects have been developed. And you have one such kind of project, which is the Saccharomyces genome deletion project, so that you can produce mutant strains in the yeast, and in which every gene, you have around 6,000 genes in the yeast, and every gene has been mutated and its mutant strain produced, to see what kind of result, what kind of anomaly, or what kind of product is being produced through the mutant strains. So this is one more thing. Now, when it comes to this, we have a prokaryote, like, what do you call it? It's not a prokaryote, I'm sorry. We have, um, please give me a moment. We have an invertebrate which is Sinorhabditis elegans. So what is this? It is a transparent nematode, belongs to the family Nematoda. So it's a roundworm. So we have different kinds of invertebrate phyla, as you might have already known. So we have sponges, then we have the jellyfish family, and then after that we have phylum Platyhelminthes, and then we have Nemathelminthes or Nematoda, and in this itself you have in Nematoda, we have around Turbellaria, Trematoda, and Cestoda. Cestodes are all almost, what do you call it? They're all parasites. And after that, you have annelids, arthropods, mollusks, and echinodermates, all belonging to your invertebrate phyla. So one such invertebrate is your nematode family belonging Sinorhabditis elegans. So it is a free living, meaning it is not a parasite. Around it's a roundworm, is around 1 millimeter in length, so you can actually see it. And at the same time, it is unsegmented, wormy form, and its symmetry is bilateral, so it's easy to see, and it has a cuticle integument, and at the same time it has four epidermal cords and a pseudocoelom. So it belongs to pseudocoelom, the pseudocoelomata, as it is known as. That is the false coelom. And this false coelom is going to, like, only the true coelom is going to cover the um, the gonads, in fact. So Coenobacteritis is bisexual. That is, it has hermaphrodites, which is both male female together, or only males. So mostly they are hermaphrodites, and males are only around 0.05% of the total population on an average. So what is a hermaphrodite? Both male and female sexual organs, that is the gonads, are present in the same organism. So you can see over here, this is the mouth, the pharynx, this is the gut, and you have the intestine over here, and this is the uterus, the female reproductive system, and the distal gonads over here. These are the spermaticae. This is the anus and the tail. So it is a transparent organism. Why is this studied in the first place? Transparent, easy for manipulation and observation. And what does it feed on? Bacteria. And it can be cultivated in a large number, that is around 1,000 worms per petri dish in the lab, and they are usually covered in agar-filled petri dishes. So you have MEM, which is easy, which is enough, and at the same time, that itself is a haven for bacteria as well. So you can study two organisms at the same time. It is a multicellular eukaryotic organism, in fact, and you can easily study cheap. It can be frozen, thawed, and it is viable even for long-term storage. And you can study cellular differentiation and developmental processes because it is a transparent work. And what is it that you're going to study in Coenobacteritis elegans? You're going to see apoptosis, which is programmed cell death. You have BCE, then BCL, then you have so many of the genes that are involved over here. So you can actually study programmed cell death, and this will help in different kinds of cancers, because usually in cancers it is the tumor suppressor genes, and when the tumor suppressor genes are mutated or there is a problem with the tumor suppressor genes, it causes cancer. or whenever there is the oncogenes that are actually getting activated, that causes cancer. So these two types of issues or can be studied through your Caenorhabditis elegans. So what is it that the researchers are trying to see with the by studying Caenorhabditis elegans in relation to cancer? So they're trying to, I hope that they can identify genes that can switch on the apoptosis in the cancer cells. Because cancer cells lose the ability to have apoptosis in them. Because of which they keep on continuously growing and cause metastasis. So researchers are trying to see some mechanisms, the neural mechanisms, wherein, the chemotaxis, thermotaxis, mechanotransduction, mating behavior, all of it, if it can be studied, then it can actually be an excellent role model for analogy with the humans. At the same time, yes, we have certain, we are trying to find basic treatment for different kinds of cancers. And yes, you can freeze thaw it for quite a long time and it is viable in the laboratory. That is a very important trait that any kind of role model organism should possess. And it is also the first multicellular organism whose genome was completely sequenced. And what is it that we're looking at? So we have a feature which is very important in the case of Caenorhabditis elegans, that is specific genes can be disrupted by RNA interference. So we have something called as small RNA interfering genes in fact. So if those RNA interfering genes are functioned, then RNA, we have that small nuclear RNAs, in fact, snRNAs as they are known as. So those, whenever you use them, they interfere with your, the gene regulation and at the same time, exon, intron, excisions, for that matter, splicing, all of it. When this is done, your protein is going to be of a different, your protein product is going to be different. So whenever RNA when you're doing silencing of gene through RNAs, small interfering RNAs as they are known as, then this will help you to understand the function of the gene in a better way. So how is it done? Caenorhabditis is soaked in a double-stranded RNA. Sequence is complementary to that of the gene that the researcher does not want to be expressed. Then this can be giving you a different product altogether. So that gene is going to be silenced. And then you have RNA interference by simply giving the worms transgenic bacteria whose RNA is going to be complementary to that of the gene of interest on which the scientist wishes to work upon. So because of this, what is happening is whenever the transgenic bacteria whose RNA is complementary to that is produced, because of which what is happening is that gene's functioning is lost. Because you have and the bacteria produces a complementary product that is the protein instead of the Caenorhabditis producing that particular gene. So that gene's function is completely lost over here. So it is the easiest in all the animals, in fact. And this is how your knockout models were produced. And 86% of the 20,000 genes in the worm. So you have around 20,000 genes present in your Caenorhabditis, of which around 86% of the genes were knocked out or silenced. So this is accounting to almost 9% of the genome that is present. So this is one thing. Please give me a moment. I would like to actually share one more slide if there's a possibility. Please give me a moment, and I would like to share one more slide if there's a possibility. According to that, we will just like see. And at the same time, I would like to talk about the different kinds of the as to why there is what do you call it the role of cytology, and at the same time the cell culture basically, and what exactly are we looking at when we talk about the cell culture. So cell culture is basically growing of the cell in different or controlled organisms. Let me share my screen with you again. Right. So, what is the necessity for us to grow the cells? To study, of course, as models again. What are we giving as a medium or as food for the cells to grow? It is growth factors, hormones, essential nutrients, which include your proteins, amino acids, and carbohydrates, vitamins, minerals. When you give amino acids, yes, that itself is sufficient for any kind of metabolism, in fact. And at the same time, you're going to give a conducible environment for the cells to grow, which is your pH buffer, the pressure, and right temperature and pressure also should be maintained. So again, we have different kinds of cultures over here, which is the artificial substrate that we are talking about, wherein the cell grows, which is known as the adherent or monolayer culture, whereas others can grow on a free floating culture medium, which is a suspension culture. What is it useful for? Different types of primary and secondary, cell lines can be grown and can be seen through different kinds of methods, such as inverted microscope, as in the case of adherent culture. Whereas in a suspension culture, you can just stay, culture it, dilute it, and then, isolate a strain. And dissociation is done by different kinds of mechanical or enzymatic methods in the case of your adherent culture. Whereas in the case of suspension culture, you do not require any kind of enzymatic or mechanical dissociation. Instead, you can just take and isolate the cell strains, in fact. Of course, for bulk production and all of that, you use a suspension culture, whereas an adherent culture is usually for research purposes. So when it comes to the brief history in the cell line as to why we are actually studying this, so you can just see that it started through hanging drop method by Ross Granville in the year 1907. In fact, he wanted to study the culture of the tissues. And from there onwards, different kinds of cell lines were introduced. Blood was used as a medium inside the medium, in fact, and different kinds of salt solutions and the concentrations were tweaked with to see what was good. And then antibiotics were used in the 1940s in the cell culture. After the discovery of penicillin and amoxicillin and all the other antibiotics, it was a different ballgame altogether. And when it comes to your carbon dioxide incubators, from 1960s onwards, it's available. Of course, today it is again a different world with so many equipment and so many things that have come up for us to study. So what is it that we're looking at? Primary culture, the cell line, finite and infinite. So primary culture is the starting stage. Basically, cells are isolated from the tissues, appropriate conditions are given, and you're putting it on a substrate. From this, many new cells are grown. From it again, you're going to isolate or subculture. That is from the primary culture, you're going to take and make it into a separate cell line altogether. So we have finite and continuous cell lines also over here, which helps in giving us according to what we need for our research. So they have, for example, fibroblasts. If you take a skin biopsy, or liver cells, that is hepatocytes, they're all from the donor tissue. They're available only for n number of times because of the telomere loss that is going to be seen after a few generations or after a few cultivation. So primary or finite lines will have a stop point, as we can call it. And if at all, the ability to divide indefinitely, the loss of what do you call it, your the um, cell control checkpoints is taking place, then you can actually get your continuous or established cell lines. This is used for long-term cells, basically long-term research. You can see them in cancer cells. That is where HeLa cells came into place. The breast cancer, the cervical cancer cells, in fact, the HeLa cells, they're still there because that loss of the checkpoints took place over there. So this is what from a cell line you can get normal, transplant, and stem cells. And where are they taken from? A tissue, normal cells, and then stem cells are those that have gained the ability to generate other types of cells. And when we talk about different types of cells, we have the epithelial-like, lymphoblast-like, and the fibroblast-like cells. Epithelial cells, flattened, polygonal in shape, and this is where these cells are form the lining of all the major organs, in fact, and there are different types of epithelium again. So according to that, lymphoblasts, like, they do not attach, but in suspension, but they are there with they're spherical with they have a spherical shape. Fibroblasts, like, that is cells are attached to a substrate, appear long, and they're bulky. And what about stem cells? Stem cells again are of different types, that is pluripotent, multipotent, and totipotent. Pluripotent is most of the cells can be developed into an entire organism. Totipotent cells can divide into all types of cells in the organism. Multipotent is one cell type you have different. So you have either lymphoid precursor cells or the myeloid progenitor cells, which are usually the pluripotent stem cells that are present. According to that, you will get it. And what are the major types of stem cells that we are looking at? Your embryonic stem cells, which are totipotent in nature. Adult stem cells have a limited amount of capacity to divide. So you can say multipotent. Induced pluripotent cells, stem cells are of another type, which can be converted into, or which can develop into any other cell that you want to. So when it comes to the different kinds of cell lines that are used in vitro culture, you have the species and the tissue of origin also has been given, and what kind of cells is going to be coming out of it. So three cell line can give is actually derived from the connective tissue, gives you a fibroblast. And CHO, HeLa cells, which are from the cervical carcinoma cells, they form your epithelial lining. And that's what all different kinds of cell lines from the tissue that are being coming out of or origin, and they give the epithelial cells. Now, what is contact inhibition when we are talking about cancer, when we are talking about different types of cells? We have certain cells that have your adhescence gap junctions. We have those whose which will actually help in arresting the cell growth when the cells come in contact with each other. So this phenomenon is known as contact inhibition. So whenever your normal cells are present, then their growth gets inhibited or stopped whenever the cell next to it is also growing and they come in touch with each other. So the cells in our body are governed by this growth control mechanisms and cellular senescence, that is aging also. So that is where your checkpoints come into place. And then you have your cancer cells. What happens is they lose the contact inhibition property, because of which they start growing and proliferating in a very uncontrolled manner. And even though when they are there with since they have lost that contact inhibition, they grow over one another, form a tumor. Now, tumors, the basic difference between tumors and cancers, if you actually take, it is because of the presence of a sheet covering the tumor cells, and it is known as the benign. And whereas when there is no sheath covering the tumor cells and it can go metastasized to different other places in the body. It is known as a malignant cells. And for malignant cells, of course, you need to undergo different kinds of treatments, be it chemotherapy or be it your radiation, all of it for that matter. Of course, along with the removal of that particular malignant cells. Whereas for tumor, basic, if you remove that particular tumor, that is sufficient usually, because the cells have not metastasized. So again, you have to maintain safety over here in the case of cell culture and the laboratory, in fact, for that matter, wearing and keeping your lab clean so that there is no amount of infection that you are incurring, or there is no release of toxins or allergic reactions. So work in a laminar airflow place. And if you're working with extremely hazardous what do you call it, organisms, say for example. So what happens is you have four different kinds of laboratories, your BSL 1, 2, 3, and 4, whereas BSL 1 is your biosafety level laboratory, that is, which is one. So basic normal or microorganisms can be worked with in the presence of a laminar airflow, in fact, always. BSL 2, BSL 3 for dangerous organisms, BSL 4 for extremely hazardous life-taking organisms such as Ebola, then Marburg virus, your COVID virus, all of which you have to work in a BSL level 4 laboratory. You have to wear the safety equipment at the same time, and see that it is completely in a very controlled manner. And of course, you have to use disinfectant, sterile medium, and you have to see everything is in place so that you do not get affected while studying about that particular organism. So, of course, filter sterilization, autoclaving is important. And you also have to take place that there is no contamination, because of which you get a different result altogether. And other contaminants usually are plasticizers that are present in the cell culture. And whenever you add antibiotics, that again risks the bacterial growth, so in the cell culture. So you can actually see how it is. If at all you see a culture, you can see these are the contaminants usually that are present, and this is how a bacteria grows, yeast, budding, and this is how a mold looks, mycelia, hyphae, all together. So what are the equipment that you use for biosafety equipment? Carbon dioxide incubator, microscopes, of course, different kinds of freezers. You have -20, and and the same time you have different kinds of freezers that are included. You have cold storage room, centrifuges, pH meters. Then you have your autoclaves, pumps, and then you have water bath. Then you have your sonicators, and of course, proper disposal of waste also should be done. So this is how your waste container should be there, right next to it. A laminar airflow probably, ethanol spray, and your equipment. Requirements, again, different kinds of media present, basal and reduced serum media, serum free media, all of which actually according to whatever requirements you have. So what do the media provide you with? The necessary nutrients, growth factors, hormones that is necessary for the cell to grow properly, and and then you have to maintain the proper pH, and also the pressure that is present. And if you're giving serum, so serum whenever given in the basal, along with the basal media, and this is what is added is usually your fetal bovine serum, calf serum, or bovine serum. Otherwise, you can use serum-free media. So you have appropriate nutrients and hormones that are given to the particular organism based on, uh, the known media basically. You know all the factors that are being given. So again, what is the basal media of inorganic salts? In pH indicator will be there, buffer is there. And also you're going to give different kinds of keto acids for your Krebs cycle and glycolysis to take place. And carbohydrate is your energy source, so you're going to give either glucose or galactose. And different kinds of vitamins are given because they are going to be a cofactor in your different kinds of mechanisms. So what are the vitamins that are given usually? B2, thiamine, that is B1, B6, biotin, and then, that's what thiamine, riboflavin, biotin. And trace elements are given, that is your micro nutrients. So you have commercial media, which is very commonly used, which is your Dalbecos MEM and your RPMI of 1640, that is RPMI as it is known as Roosevelt Park Memorial Institute. And you have an F12 media, which is given. So based on this, different kinds of growth of different organisms can take place. And again, you're going to maintain the physiological pH of around 7.2 to 7.4, and carbon dioxide is given. Temperature is maintained for optimal growth of any cell medium. You have to maintain an optimal growth. And of course, cold-blooded creatures tolerate a wide temperature fluctuation. That is why they're known as poikilotherms. Whereas for homeotherms, then it is like only up to 36 to 37, which is optimal in nature. So what is it necessary for? Basically, how are you going to choose cells for application? So we have already seen some certain modeled organisms over here. Now, when you choose for your different kind of study, what is it that you're seeing? Cell lines, and what is it that you're seeing for? For what kind of study are you seeing that? Is it for toxicity studies? Then you're going to see liver and kidney derived cell lines. That is because liver and kidney are the most important detoxification organs in the body. Liver detoxifies, and also at the same time, what do you call it? It helps in all metabolism. Kidney is your primary blood filtering organ. So mostly for toxicity studies, you're going to see your kidney and liver derived cell lines. And for cloning and for different kinds of cell studies, you're going to see your cell lines. And when if it is normal or transformed, then you're going to see depending upon that, the organism can be chosen. Growth conditions also have to be taken into consideration, especially for bacteria or yeast, and what kind of cell that you want. So in vitro, if at all you see cell cultures, then you can see that frozen cell population can be revived through different kinds of from the tissue. But continuous freeze thawing is going to cause the cell lysis. So you have to take care of that and give it optimal temperature before you thaw it and before you use the cells and study. So we have batch culture again, old, new, then cell passage or subculturing. And according to this, you can actually see your lag phase. This is your growth curve basically and growth pattern. Lag, exponential or log phase, stationary phase or the plateau phase, and the death phase. And transformation is wherein, whenever there is a alteration in the growth rate, mode, if there is any tumorogenicity and mutations, and if at all there is any chromosomal differences like indels, all of it can be seen, and it is associated with the cell lines, wherein you can actually immortalize the cell line. So because of this, indefinite proliferation is taking place in vitro in your test tube or in the lab. This will actually be useful in the cell cycle. And so what is it going to do? It stops or the expression of the gene which is encoding your telomerase. And this can be done through inclusion of viral genes such as SV40, simian virus 40 large T antigen gene, and telomerase gene HTRT. All of this can be used. And aberrant growth control, it helps in reducing the anchorage dependence so that the cells can grow in a suspended culture. And where is it used? For overexpression of certain oncogenes, or if at all you have to inactivate tumor suppressor genes or growth suppressor genes. And tumorigenicity is the ability of the transformed cell to see that it produces invasive tumors which are malignant. That is, they are they will change the growth control pattern, become disease causing or malignant. How are you going to maintain these cells for studying whenever you're doing? By cryopreservation. That is, after you harvest it, you use a culture which is around, which is DMSO, which is a cryopreservant, around 10% of it. This is added so that, it minimizes the injury to the cells during freezing and thawing. Also, if at all there is ice crystal formation that is taking place, because of which what happens is the cell is going to burst in ice. We know that at 4 degrees Celsius to 0 degrees Celsius, or whenever there is water is being frozen, it to form ice, its volume increases. If this happens in the cell volume increases upon thawing, or upon freezing and thawing, because of which the cell is going to burst out. So to avoid this, you use 10% DMSO, which is your dimethyl sulfoxide. And then at the same time, you're going to take it, transfer this to be frozen at minus 80 degrees Celsius in another liquid nitrogen storage tank, wherein liquid nitrogen can actually, is present. The nitrogen is, the temperature of it is around minus 196 degrees Celsius, wherein the nitrogen stays in the liquid form. So it is the best way to preserve any kind of, organism, in fact, and or tissues for that matter. And then once this is done, you have cell counting. So to know the number of cells that are present, you use an instrument called hemocytometer. And then you use a dye, and then with the help of a cover slip, you're going to actually count and then extrapolate it. And that is, you can calculate the concentration. So there is a standard formula and a protocol to do that, basically. Now, how are we going to see if at all there is any kind of issues or we are facing, basically? What is the problem that we are going to face, and what is the reason and the solution that can be seen? Because these are the basic common lab issues that we are going to face. One is number of viable cells as soon as we thaw the frozen samples. So what is the problem with it? Incorrect storage or incorrect thawing. So according to that, you have to see that the only thing that can be done is obtain a new stock. And that has been in the liquid nitrogen. So that is why we need to aliquot the samples and store many samples of the same strain, so that, in case there is an issue with thawing and you do not get viable cells, you have other samples to work upon with. Incorrect thawing also, that will actually cause issues. And so dilute the frozen cells slowly with pre-warmed medium, and then centrifuge at low speeds. And if at all there is glycerol exposure to light also, this can happen because of no the lack of viable cells can be seen. So because of which, what is happening is, if at all glycerol exposure to light is happening, and you're adding glycerol as the cryopreservative instead of DMSO, and it's been exposed to light, which is not good. And then acrolein is a product that is going to be formed, which is going to be toxic to the cell, because of which your viable cell count has come down. And if at all there is any lack of attachment to the culture vessel after subculturing, so that is because of any kind of the digestive enzymes that are present, and cell adherents are working, are not working properly, or because of any mycoplasma contamination. Based on this, you have to see either you have to give the medium for replating, or you have to block the enzymatic activity using an inhibitor such as FBS, fetal bovine serum, for trypsin, and or else you have to perform a test for your PCR test to see whether there is any kind of mycoplasma, what do you call it, contamination. Slow cell growth can be because of incorrect growth medium, depletion or breakdown of essential cell culture components. So this again requires different kinds of growth medium that has to be given properly, and then keep looking for different kinds of amino acids and different kinds of substances, and like such, like look for glutamine with glutamax, etc. And what are the applications of the cell culture? The culture, you see, it is used as a model culture for basically studying your cell and cell biology, biochemistry, in fact, research, and at the same time for studying different kinds of or like what do you call it, diseases, and to studying drug toxicity, to study senescence, to study uh, like you know uh, and for cancer research, that is, you're going to see the difference between your normal cell and cancer cell, and at the same time you can see how normal cells are being converted into the malignant or cancer cells through different kinds of methods, be it physical, chemical, or biological. That is, physical is through what do you call it? Uh, b- b- if at all there's any kind of radiation, in fact, that is being given, and chemical is if at all you give any kind of carcinogenic agents, then will it cause EDTA is one, and then your DMSO is another carcinogenic for that matter, and ethylene bromide is one. I'm sorry, not EDTA, but it is ETBR, ethylene bromide. And you have so many carcinogenic chemicals, in fact, for that matter. And then viruses. Different viruses can actually cause cancer because of their ability to go and attack the cell control, your tumor suppressor genes and your or activate your oncogenes. So cancer mechanism can be studied, and then at the same time, drug delivery system. What is the drug that can actually work upon? That can be studied, and at the same time, virology. Virology is a huge topic right now, and whenever animal cell cultures are used to replicate the virals, this is the viruses. In fact, you can see the study of vaccines. There are so many viruses whose vaccines we do not have. The most famous and common example that you get to see is that of the HIV virus. We do not have a vaccine against it. Malaria. For malaria, we do not have a vaccine. For dengue virus, for dengue is caused by virus, in fact, and the organism that is going to actually transfer this particular virus is Aedes aegypti, which is your dengue mosquito. So many of them are there, and you do not have a vaccine, you do not have treatment for Ebola, Marburg. So many of them, those do not have medicines, treatments, vaccinations, nor do you have any kind of prophylactic or preventive medicines. So yes, cell culture can be used to detect, isolate the virus, study the cell growth development, so that you can study the mode of information in infection, and at the same time, hopefully, look for different kinds of medicines against them. Vaccine production, again, for vaccines for polio, rabies, chickenpox, all of them were produced through your animal cell culture. And now we have an issue with production of whole organ or tissues or organs. So far, no particular organ or tissue could be produced successfully for treating patients with issues. So probably artificial skin for burn victims, ulcers can be done. So far, what has been done is take skin grafting from other part of the body to be put onto the burnt part. That has been done. But and at the same time, transplantation. Uh, it's like cadaver, that is your dead body, to the recipient has been done so far. But then, apart from that, you do not have a successful transplant that is being done. Those people who do not have, especially in heart transplantations, There's not been a successful heart transplantation that was done. So couple of years ago, what has been done is pig heart to human heart. But unfortunately, not more than a month or 2 months, the recipient could survive. And genetic engineering, of course, new material like DNA or RNA into the cell is being incorporated. Usually, through genetic engineering, you're producing different kinds of hormones, insulin, and for usually what is happening is this is infecting them with baculoviruses, and then production of the hormone or protein that is being done. So expression of the genes. Gene therapy. Gene therapy is wherein, what is happening is through this you are trying to use or see that when you are tweaking or removing that gene which is not functioning properly, or if that functional gene is being missed in a patient, then you're culturing that particular gene and introducing these genes into the patient when at the genome level, so that because of the gene that is present in the patient now, it should be able to actually help in the production of protein, or that gene is becoming functional. So a maximum they're trying to do in cystic fibrosis, basically gene therapy in fact. And again, it is not going to work all the time. And in case of different kinds of muscle issues, there is a gene therapy which works. In fact, the most expensive drug in the whole world, which is costing you around 16 crores, is a gene therapy drug that is being used, in fact, to treat your muscle issues, and that can be only done when the baby is say less than 1 year or 2 years max, or 1 year or 2 years. Otherwise it is not going to work. So yes, there have been they're trying to do a lot of gene therapy as well. And when it comes to that, we have even screening development to learn about the toxicity of drugs, and to see if there are any kind of new drugs that are available, and what are the side effects of those drugs as well. So for all of these, you need cell, different kinds of cell and cell culture that is being done. Now, I want to actually just go through the difference between your the cytogenetics and your cytology, and how it has been interlinked together, and what is the chromosomal basis for inheritance. What should we know? So before that, what can we happen? What is a genotype and what is a phenotype? And then why are we having different kinds of disorders? All of it put together, let us just have a look at it. So when it comes to that, how many chromosomes are in the cell that are diploid, triploid, or what is it basically? What do we know when we study about cytology and genetics, and why is it all mixed up for us? So we just see the chromosomal theory of inheritance. So what should we know about it? Please give me a moment. And the necessity to basically link the genes, and why has cytology been linked with your genetics as well. So when we see that genes are present on the chromosomes, and these chromosomes are going to be segregated and independently assorted. So it was Thomas Hunt Morgan, in fact, who worked on Drosophila melanogaster, and the Drosophila melanogaster is the fruit fly, and it is basically the chromosomes that are going to cause or talk about the inheritance basically. So again, you have different kinds of inheritance over here. It can be the XY system, XO system, or the ZW, or the haplodiploid system. So you have a lot of sex-linked genes that are present on either the chromosomes X or Y. So And according to that, you have disorders also such as color blindness and Duchenne muscular dystrophy, or hemophilia. And of course, you have and over here they have just given you how hemophilia has come into it's also known as the royal disease because it was actually identified and it is very prominent in the royals. And when it comes to inactivation of any kind of chromosomes, you see that bar body is the inactive X chromosome that is going to be present, regulates the gene dosage in the females. In the case of human development, so you know that chromosome has two arms, and the P and the Q arm with the centromere over here, and you have the telomeres over here. So sex determining region is the SRY. This is the sex determinant region Y gene, and it's the short arm and the long arm. This is the Y chromosome. And this is going to be found only in the Y chromosome, because of which it codes for protein that regulates the other genes. So recombination will have you the offspring. If it looks like the parents, it's known as the parental type. Different from the parents, then it is a recombinant. So basic. So linked genes are dihybrid female and homozygous recessive male. When you do a test cross, you're going to get the results as such. And crossing over actually explains some linked genes that get separated during meiosis. Your meiosis 1 produces the crossing over and frequency, recombination. Meiosis 2 is basically mitosis. So you're just going to get the recombinant chromosomes and doubling of them, which is going to take place. And through this, you can actually calculate the recombinant frequency as well. And you have something called as the linkage map, that is the genetic map, which actually tells you the percentage of the crossover event that is taking place, wherein one map unit is 1% recombination frequency. This also gives you the relative distance between each chromosome or the genes along the chromosome. So if at all there is 50% recombination, on the same chromosome, you have the recombination taking place or on two different chromosomes. So again, exceptions here, exceptions are present. That is the genomic imprinting. That is the phenotypic effect of the gene depends upon whether it is from the male or the female parent. And then you have methylation, wherein, whenever you add a methyl group to the DNA, then it causes the allele whenever that is present. If it is inherited from the mother, then you're going to get a wild type normal sized mouse, and then the IGF2 allele is going to be expressed. And then when you do this, in the case of mutation, methylation, if it is done, then the IGF2 allele is not being expressed. If the mutant IGF2 allele is being inherited from the father, you get a dwarf mouse, and then the allele is expressed, and normal IGF2 allele is not expressed over here. You have certain DNA, which is the non-nuclear DNA, which is present in mitochondria, chloroplasts, plastids, and then you have the, what do you call it, bacteria have plasmids, which have the small circular DNA that is present. So mitochondria is your maternal inheritance. We have seen different kinds of diseases also pertaining to it. And all of it are maternally inherited. And so what is it that we are going to see through this? What is it that we are going to do? Why is genetic, cytogenetics very important branch of biology altogether? That is because your genetic disorders, they're inherited, and you're going to do some testing for that, and you need predominantly cytology is very important. Pre-symptomatic predictive testing can be done because of different kinds of genes. If at all they're inherited, you for sure know that yes, it is going to happen. So one of them is your Rh negative positive, and if it is an Rh negative father, Rh positive mother, and yes, you're going to get the baby might, the first baby might survive, but the second, third, fourth issues are not going to survive. And you have to see that the the what do you call it, the mother takes exclusive care so that, what do you call it, the you do not have any kind of miscarriage. Carrier testing before having children, yes, you can get genetic counseling done. And then pharmacogenetics, medication dosage, prenatal, antenatal, that is newborn screening is antenatal screening. Pre-implantation testing also is done. Prenatal testing is when the female is pregnant. That is when you can actually detect certain genetic disorders. So, in the third month or the first trimester itself, you have a certain, certain tests that are done, which can actually detect different kind of genetic disorders such as amniocentesis and chorionic villus sampling, which is your amniotic fetuses taken and karyotyped. And then uh, uh, the sample of placenta is taken and along with the fetal cells and karyotyped. This actually gives you, after karyotyping, if at all there is any kind of genetic issue, you will be knowing it. And non-disjunction of chromosomes is whenever there is any kind of failure to, what do you call it? Just give me So whenever there is no a failure of the chromosomes to actually separate, it causes non-disjunction, and because of which it will be the anomalous anomaly in the chromosomes. So because of which you get monosomy, trisomy. Usually because of non-disjunction you get trisomy 21, it is Down syndrome. Trisomy 18, Klein, then and then you have Klinefelter syndrome, wherein you have the 47XYY or 47XXY, the non-disjunction in the sex chromosomes. Turner syndrome 45XO. So you do not have another either the Y chromosome or the X chromosome over here. So it's a monosome. So you have the mutations which are deletion or the duplication of the chromosome. In fact, inversion wherein the chromosomal segment is getting inverted, or translocation. And because of non-disjunction, you have aneuploidy, which is monosomy one copy, or trisomy, which is three copies. Polyploidy, which is like two, that is rare in usually polyploids are seen in plants. And tetraploid animals are also there. Again, this is because of errors in mitosis or meiosis. So these are the kinds of issues that can arise, because of which we study cytogenetics altogether, and it is a completely different branch. So I'd like to stop sharing over here. Now I would like to talk about different the basics introduction of transposons and transposable elements, which I would like to talk about again in a much more elaborate manner. But before that, I would like to share one video first. Please give me a moment. I would like to share one more video with you. Please give me a moment. I'm actually looking for the slide right now. And I actually have to talk about one particular organism, which is Drosophila, in fact, and why it is used as a model organism, especially in studying, and how important it is. So just give me a moment. I will just like download that particular. I will open that particular slide. Yeah, share screen. Right. So when it comes to Drosophila melanogaster and why it is an important organism for life sciences, you get to see it is a very important organism because it has been exclusively and extensively studied for more than a century, in fact. Why? Because its features are very similar to that of the human beings, and approximately 60% have been identified, and they can be amplified, deleted, inserted, and you have according to the different kinds of diseases that are present. And also it has a short lifespan for that matter, and number easy to culture, low number of chromosomes, small genome size, and at the same time you can easily manipulate it. So Thomas Hunt Morgan in the year 1909, in fact, he along with different students, Calvin Bridge, uh, Herman Muller, all of them, they had actually studied on Drosophila, and according through this only they could actually explain your sex linkage. Then you have your gene bridge balance. In fact, Calvin Bridges was one of the scientists, in fact, and they could give that they could talk about the sex linkage, and genes contain chromosomes, demonstration that genes are arranged in a linear order with a fixed measure between them. All of this could be understood by mapping the Drosophila genome. So, what is it that is important in Drosophila? So, its life cycle is quite small, in fact, just around 12 days to complete at room temperature, which is 25 degrees Celsius. And it is quite easy to actually look at the different stages of it, since it is a fruit fly and insect. So you have the egg, larval, pupal, and adult form. So through this, you can actually study the different phases of it. And during metamorphosis, it is going to emerge as an adult fly after following different kinds of larvae and pupal in start stages. Now, hundreds of these embryos are collected, and then the material can actually be stored in liquid nitrogen, and you can actually add RNA called 1, 2, or different proteins such as histones for chromatin structure analysis. Also, when it comes to the genetic structure, it is not at all complex. In fact, it has around 8 genes, and proteins, histones, they are how they are packed in the cell's nucleus. And over here, you have only 4 pairs of chromosomes. So, two X chromosomes, one X, one Y for the male, and they are along with chromosome 1. And then you have 3 pairs of autosomes, so 4 chromosomes. And fourth chromosome is the smallest and is called as a dot chromosome. So with 4 chromosomes, you can easily understand it, and you can actually do the genetic mapping and understand the fruit fly. And so red eyes are normal or wild type in the Drosophila, Mutant strains have white eyes. So again, this is because of the different kinds of experiments you can actually find, because of which they could find the chromosomal inheritance, in fact. So why is it used as a model organism in terms of base pairs? You have around, it's only around 5% of the human genome size. And it has 132 million base pairs for the fly compared with the 3.2 billion base pairs that are present in the human beings. So easy to study 132 or 13 lakh base pairs, in fact, as compared to your 3.2 billion base pairs. Also, around only 15,500 genes are present on the four chromosomes, whereas we have around 22,000 genes that are present on 23 chromosomes. So, density of the genes per chromosome is more over here because 15,500 genes are concentrated on those four chromosomes, whereas for us, the density of the chromosomes is varied. It is less for us because 22,000 genes are on 23 chromosomes. So if you take it nearly as 16,000, you have 4,000 genes on every chromosome. Around 3,900 genes are present on each chromosome, whereas for us, it is a different matter. It's around 1,000 genes on each chromosome. And in Drosophila, it is higher than that of the human genome. You can just see. And same genes have been maintained from through the homologous for us. So around 60% of the genome is the same for us. And approximately 60% of the genes associated with human cancers have been identified with that of the Drosophila genome also. So cancer genetics is an important role in which Drosophila is a role model. When it comes to the mechanism to equalize the dosage of the X and the Y chromosome, you can actually inactivate the X chromosomes in the mammals. In Drosophila, dosage compensation can be twofold by upregulation of the male X chromosome also. And dosage compensation in Drosophila increases the transcription of the genes on the single X chromosome that is present on the male, unequal to that of both X chromosomes in the females. And if at all you want to do histone acetylation by male specific lethal complex, this is going to increase the transcriptional output in the males, X. And in case in the humans, male cells have a single X chromosome and we have a Y chromosome in case of humans. So in Drosophila also it is the same. Females have two X chromosomes. So, researchers have actually seen how the X and the autosomal chromosome gene expression is being equalized between both the sexes, and this is known as your activation model and the inverse model. And the study about the male specific lethal complex functions, so which is going to be a regulatory protein complex, sees that there is a gene expression control in the male Drosophila or the male fruit fly cells. This activation shows that MSL actually upregulates your transcription of the X-linked genes twofold in the male cells. And it comes to inverse dosage effect, sees that the males actually, the MSL represents the male autosomal gene expression so that there is a genic balance theory. So this is your genic balance theory that was proposed by Thomas Hunt Morgan student Bridges. And molecular mechanisms of X chromosome inactivation also can be seen. In fact, that is cis RNA is established, upregulated, and inactivation of the X chromosome that is coated with your stabilized cis RNA is done. And then once the X chromosome is silenced, and then you have the RNA coating, you then this mechanism is not yet known, and then you can actually study this mechanism of the X chromosome inactivation. And then you can modify the chromatin. You can do histone deacetylation, methylation, and different. This is done on the histones or the promoters of the X-linked genes or the transcription factors for that matter. Or this is one of the reasons why your, what do you call it? Um, uh, your Drosophila is used as an excellent model for different kinds of cytogenetic studies. Yeah, I am going to stop my class over here right now. Tomorrow we will just have a look at the topics, that is your transposons, and as well as a recap of the cancer genetics. That's all for today. Thank you so much.